Yes, sir.
So I was telling you about different methods of streaking. In that, I was telling you about quadrant streaking. Quadrant streaking. So this is the quadrant streaking. Next, 3 times streaking, 3 times. So then another method is, this is also called as a T method of streaking. So here, so instead of 4 streaking, we will be doing 3 streaking, but this is a little bit different from a triangular streaking. So here we will divide the petri plate into 2 parts. First is into 2 halves. The second half will be again divided into another half, 2 halves. So here initially you take the inoculation inoculum. After sterilization of inoculation loop, touch the colony, do a one long streak, which will cover an entire half plate. Again, sterilize the loop without touching the colony. Need not to touch the colony. So by touching the first line, you just do another close streaking. Second time, again sterilize the loop and do a third streaking by touching the second streak line. So do a loose streaking. So this is called as a T method of streaking. It will look like a T. You can see here the division of the plate. Okay, like this. This is the first streaking. I'm sorry. So this is the first streaking. So this is the second streaking after sterilization of the loop by touching the first streaked line. And this is the third one, loose streaking. This is T method of streaking, like this, exactly like this. So take a inoculation loop, sterilize, touch the colony. So do a first streaking. So almost till the half of the plate, turn the plate. So sterilize the inoculation loop, touch the first line, do a second streaking, again sterilize. By touching the second line, do a loose streaking. So this is the another method of T method of streaking. Okay. So these are the major method of pure culturing streaking methods which are followed. Here, the one doubt which you may get is, why we need to do, why can't we do a simple streaking? Just take the inoculation loop, just do the one simple streaking. So why we are doing 3 times, 4 times, and why we are sterilizing each times? You may ask the questions. So ಇಲ್ಲಿ ನೋಡಬಹುದು. This is 3 times. So here, why we are doing 4 times streaking is, so when you take a inoculum from this colony, this contains huge number of cells, which contains huge number of cells, crores of cells. So even when you put an inoculation loop, it will attach lakhs of cells. But when we are pure culturing, we need to get a colony. Again, when you are pure culturing after streaking, we need to get a single colony that is coming from a single cell. We need to get a single colony that is coming from a single cell. So here, so there are chances that the more cells will be there at initially in the inoculation loop. So why we are doing so much streaking means, so we are reducing the number of cells. We are reducing the, each time by streaking, we are reducing the number of cells that are attached to the inoculation loop. So after first streaking, again we will sterilize by, and we will kill all the cells that are present in the inoculation loop. We just touch the diluted cells here and we will do a streaking again. So in all these methods, we are trying to reduce the load that is there on the inoculation loop. And we are trying to put somewhere, so individual colonies. So there should be individual cells should be there, so that the colonies which are coming here, so they are coming from an individual cell, not from 1 or 2 cells. So if there are any contamination, even if there are any contamination, little bit contamination here, so they will be get eliminated in these streaks. And if the isolated colonies which are coming here, they are pure in them. So here we are expecting the isolated colonies. Here there is a continuous growth. So you can see here there is a continuous growth. So from here, third streak, the isolated colonies have started. So again, from this you can take and streak it on the, do a simple streaking on the second plate, you will get a pure culture of that organism. Similarly, here you can see. So there is a more number, because of more number of cells, there is a continuous growth. This cannot be taken for pure culture. So here also there is a continuous. So from third streaking, so you have got a isolated colonies. These are the pure cultures coming from single cells. So this we will take up and put it on the agar slant. Yesterday I have explained to you about the agar slant preparation. So take this, stick it on the agar slant, allow it to grow and store it. That will be a pure culture. Here also you can see. So initially, so there are a lot of number of cells, continuous growth is there. So at the end, you can see the isolated colonies. From this, you have to take this and put it on this land, and you have to grow it and maintain it as a pure culture. So all these streaking we are trying to reduce the number of cells or load that are there on the inoculation loop, so that we will eliminate if there are contaminants, any contaminant, we will get a isolated colonies from single cells, which can be so again transferred using a sterilized loop onto the slants, agar slants, grow them, label them and store them as a pure culture. Same thing here, T method of streaking. So you got isolated colonies here. Okay. Now, so these are agar slant. Yesterday I have told you how to prepare a agar slant. So these are the agar slant. So you can just the isolated colony which are coming here, you can take this, you can streak them here. So allow it to grow for 5 to 6 days. After growing, so you can label it and store it in a refrigerator at 4 degrees centigrade, 0 to 4 degrees centigrade. Usually we will store. So next one is, once you get a pure culture, once you get the pure cultures, one second. So I will go back to the yesterday's, sorry. Yes, yeah, I'm going back to yesterday's pictures. Plants are not there here. I don't know where I put that. Yes. Sorry, I'm not getting those. One second. I'll show you here. Pure culture. I just wanted to show a pure culture on agar slant. So from this isolated colony, if you can able to see here the pictures. So from the isolated colony, whatever we got after streaking, this will be streaked down to slants, agar slants. I'm enlarging. So from these colonies, so after streaking, we will be streaking onto agar slants. So we will grow them, label it, and store it in a refrigerator, 0 to 4 degrees centigrade. These are the agar slants. So where we store the pure cultures. Okay. So then the preservation of pure cultures. So once you can able to do a serial dilution, you get a isolated colony, streak them, you get a isolated colonies which are pure in form, then streak it onto this land. So what are the different methods of storing? So ಯಾವ method ಅಲ್ಲಿ long term store ಮಾಡಬಹುದು. One is just by preparation of the agar slant. So you can store them 0 to 4 degrees centigrade under refrigerator. So whenever you want a pure culture for any purpose, you just take this slide. Just take this slant containing a required pure culture. So in a laminar airflow, using a sterile inoculation loop, take a little culture by just touch it, stick it on the new agar plate, new 10 media plate, you will get the growth of that culture. From this, you can put it into broth or anywhere, whatever the purpose you want, you can use them. So these lands can be stored up to 6 months. They cannot be stored for years together. So why they cannot be stored means, so in a refrigerator where there is 4 to 6 degrees centigrade, although the growth of the microorganism and metabolic activities are greatly reduced, but still there is a slow rate of growth. Under refrigerated condition also, there will be a very slow rate of growth. So because of which, so there will be nutrient depletion, continuous slow nutrient depletion will happen. So it will dry up, the media will dry up, and also the cells will be dying with because of with the, growth curve, I think. So with the time, these cells will be dying. So there will also be accumulation of toxic materials because of all these. So it is better to change from one slant. So you have to change it to next slant every 6 months to maintain a pure culture. From this slant, again stick it onto new plate. From this plate, you stick it onto another new tube, grow it, store it for another 6 months. So validity of these slants are 6 months. So is there any other methods where we can store this for a longer period of time? Because it is a very time consuming. If you have a hundreds of thousands of cultures, so every 6 months if you want to do means, so it will take another 1 or 2 months for this purpose itself. So are there any methods where we can store for long period? Yes, there is a method. So here, so this tube where you have grown a bacterial culture or a fungal culture, so if you can able to pour a sterile vegetable oil, any sterile vegetable oil, or to better liquid paraffin. So liquid paraffin, if you fill this until the mouth, so what will happen? There will not be, if you fill this with a liquid paraffin or a sterile oil, so there will not be entry of air. Air will not enter inside. So and also the metabolic activity will be fully stopped. The activity of the organism will be fully stopped, and they will not be utilized because of no aerobic condition. They will go to dormant state. So there will not be utilization of these media. So all organism will go to dormant state. So in this state, we can store the culture, pure culture, for up to 1 year. We can store the pure culture up to 1 year. Okay. So there is another method where you can store even longer, even longer. Cryopreservation. Cryopreservation is, so whatever the slant you have, so you should prepare this culture in a cryo vials. Huh? That slant in cryo vials means which can withstand a very low temperature. Cryo vials. So this type of tubes will, these are the cryo vials. Plastic tubes will come which can withstand the very low temperature. So what we do, whatever the slants are there, we will be freezing them at a very faster rate. Immediate freezing will be done at minus 190 degrees centigrade. So the liquid nitrogen, temperature of liquid nitrogen is minus 190 degrees centigrade. If you just dip that, it will suddenly freezes. All the metabolic activity of the cells, everything, it get freeze suddenly. So after freezing the pure culture in liquid nitrogen, store it in minus 80 degrees centigrade. There will be a deep freezer which is available, which will maintain a minus 80 degrees centigrade. That culture which is frozen will be stored in minus 80 degrees centigrade. Whenever you want the culture back, so here the cultures will be stored in multiples. At least minimum 5 set will be there in this freeze culturing. So what we'll do is in freeze culturing, here one more precaution while doing a freeze culturing is, so you have to add the protectants which will not the cell stabilizers, which are called as a cell stabilizers, so which will protect cell from forming a ice. So if the cell content forms a ice, so it will granulate and it will break the cell organelles and it will kill the bacterial cells or fungal cells. To avoid that one, before freezing, so this culture we will not take the agar slat. We will take a liquid culture. We will take a, please listen, we will take a, while cryopreservation, we will take a liquid culture broth, liquid culture broth which is grown. So to this we add a stabilizer, cell stabilizer. Usually we add glycerol. Glycerol at the rate of 20%. Usually 20 to 30, 10 to 30% you can add. Standard is 20%. So you add, so here 20% means So every 40 ml culture, you add 10 ml glycerol, or every 80 ml culture, you add 20 ml glycerol, mix it vortexes, then freeze it suddenly by dipping it in a liquid nitrogen, store it in a deep freezer. Storing cultures in deep freezers. So -80 deep freezer will be there, -20 deep freezer will be there. These are the deep freezer, -80 deep freezers. Store it. So in this, you can store for many years. So these are also called as glycerol stocks. Glycerol, because we will put a glycerol. So it will be in the form of ice itself. It will be suddenly frozen, but cell inside the cell, the liquid will not be frozen. So whenever you want pure culture back, take out one tube. So one tube, before thawing it, before it becomes a liquid again in the room temperature, scrape the pure culture little bit, stick it on the new media, and you will grow that one. So once it become thaw, it cannot use, but the cell will get killed and you cannot reuse that one. Okay. So another method is called as a lyophilization. In this method, what we do is, in lyophilization, what we do is, this is called as an instrument which is called as a lyophilizer. So which will suck it is done from, pure culture is done from liquid culture, liquid broth of fungi or a bacteria. So this liquid broth of fungi or bacteria is frozen. Here it is made frozen by putting in a -80 or dipping it in a liquid nitrogen. Then it is connected to a lyophilizer, which is at -90 degrees centigrade, -80 to -40 degrees centigrade. So since the temperature is very low, -90 to -40, once the culture becomes ice, frozen, here also you need to have you need to add a stabilizer. Once it is frozen, when it is in ice form itself, by process of sublimation from ice itself, it will remove the water. It will remove the water content from the bacterial cells and other media, and it will become a powder. So this powder can be stored under room temperature anywhere. Whatever the temperature you want, room temperature, you can store for many years. There are instances that it will be viable even for more than 20 years. These culture lyophilized powders are viable for more than 20 years. Okay. So these are some of the methods of preservation of pure cultures. Okay. So this is what I wanted to cover in this class. So if anybody has any doubt, you can ask me.